Welcome to the Deep Dive. Today we're tackling a really fundamental topic in immunology, peripheral immune tolerance. It's core stuff and very timely. It's the basis for the 2025 Nobel Prize in Physiology or Medicine.
Exactly. It's a fascinating area. Our mission today really is to unpack those specific Nobel-recognized discoveries. We're talking about the work of Mary Brunko, Fred Ramsdell, and Shimon Sakaguchi. They essentially cracked the code on the immune system's critical backup plan.
Okay, so before we dig into the specific genes and, the researchers, let's set the stage for you, our listener.
Yeah.
What exactly is peripheral immune tolerance? And why call it a backup plan? What's plan A?
Right. So plan A is central tolerance. That happens in the thymus. It's like the immune system's boot camp. The body tries to weed out T cells that react against your own tissues, self-reactive cells. But, well, it's not perfect.
How imperfect are we talking?
It's maybe only 70% efficient. So quite a few potentially problematic T cells and B cells, too. They slip through. They escape into the bloodstream and tissues, the periphery. And that's where peripheral tolerance comes in. It's the second line of defense out in the field to stop those escapees causing autoimmune disease.
Okay, so it stops self-attack.
Mm-hmm.
But it has another job, too, doesn't it? It helps us ignore things that aren't dangerous.
Precisely. It prevents unwanted reactions to harmless things we encounter every day. Think food antigens, maybe common allergens, the bacteria in our gut. It keeps the peace. And the Nobel work really zeroed in on how the body actively enforces that peace outside the thymus, finding the molecular controls.
So what was that key discovery? The molecule that kind of blew the field open, I remember reading it, was linked to a pretty severe disease model early on.
Yes, that leads us straight to Mary Brinkow's work. She was instrumental in co-identifying the gene we now call FOXP3. Back then it was called scurfin. And the critical link was to the scurfy mouse. These mice have a mutation, and they develop this fatal lymphoproliferative disorder, basically. Their immune system goes wild. Brunkow's work showed that losing this one transcription factor, FOXP3, is what drives that uncontrolled T-cell activation and lethal disease.
Wow. So finding FOXP3 basically pinpointed it as the master regulator, the key instruction manual for keeping things under control. It essentially defined the function of this whole cell type, the regulatory T cells, or TREGs, right? They are the cells that express FOXP3 and actively suppress immune responses.
That's exactly it. FOXP3 is the lineage-defining transcription factor for TREGs. Identifying it gave us the molecular handle on these crucial suppressor cells. It provided that genetic foundation.
And knowing the gene is so powerful, like we saw with Brunkow's other work in bone genetics with SOS sclerostin, find the gene, understand the pathway, and maybe you find a new way to treat disease.
Absolutely. That genetic clarity is often the key.
Okay, so FOXP3 directs the TREGs, the Peacekeepers. How do they actually do it? What are the main ways T cells are kept in check out there in the periphery? I think you mentioned 4 main intrinsic mechanisms.
Yes, 4 main ways antigen-specific T cells are controlled intrinsically. First, quiescence. T cells leave the thymus in a resting state, G0. This stops them from getting activated just by background noise, tonic signaling.
Okay, so they start off quiet.
Right. Second is ignorance. Sometimes a self-reactive T cell just doesn't see its target antigen properly. Maybe the concentration is too low, or the antigen is physically hidden away in places like the eyes or the brain, what we call immunoprivileged sites.
So sometimes they just miss each other. What about when they do meet?
That brings us to anergy. This is really important. It means functional unresponsiveness. A T cell might recognize its antigen, that's signal 1, but if it doesn't get a second signal, a costimulatory signal, usually from an antigen-presenting cell, it gets shut down. It doesn't die necessarily, but it becomes unable to mount an attack or produce the usual effector molecules. It's silenced.
Silenced. And the last one, to the really dangerous ones.
That's peripheral deletion, basically getting rid of them. Self-reactive T cells that manage to get activated despite other checks can be pushed to undergo programmed cell death, apoptosis. The BIM pathway is one key route for this.
And it's not just the T cells acting alone, right? You mentioned other cells helping out.
Correct. There are other important players. Certain types of dendritic cells, we call them tolerogenic DCs, can induce energy or even help convert regular T cells into TREGs. And even structural cells in the lymph node stromal cells, or LNSCs, they play a role in presenting self-antigens in a way that promotes tolerance, not activation.
So it's a whole system working together. Pulling it back then, identifying FOXP3 was really the breakthrough moment. It gave us the molecular basis for understanding how the immune system actively polices itself.
It did. And it connects to other concepts too, like split tolerance. If you tolerize the T cells, you often prevent B cells from making autoantibodies because B cells usually need T cell help to get fully activated.
Right. So the big takeaway is that understanding FOXP3 and TREGs gave us this incredible insight into the active mechanisms the body uses to prevent self-destruction.
Exactly. And that, of course, leads to the really exciting question moving forward, the provocative thought for you to consider. Now that we understand these natural control mechanisms, FOXP3, TREGs, energy deletion so much better, how can we manipulate them? How can we leverage these pathways for new therapies? Not just for autoimmune diseases, which is the obvious link, but potentially beyond. We saw with Brunkhau's work on SOSD how targeting one molecule led to anti-sclerostin therapy for bone disease. Could boosting tolerance pathways, maybe in specific ways, offer treatments for conditions we don't currently think of as purely immune-related? That's the frontier. What else can we achieve by fine-tuning this natural system of control? Something to think about.